everyone, sexual reproduction in flowering plants from this chapter subtopic seed and fruits. The seeds and the fruits are the structure that develop after the fertilization, right? So after the fertilization, the ovule that develop into seed and the ovary that develop into fruit. So the seed often described as the fertilized ovule. So these seeds are formed inside the fruits. The seeds which are present or they are formed inside the fruits. So let's see the structure of the seed. The seed, the typical seed, mainly it contains the seed coat and the cotyledon and the embryonal axis. So these are the major important parts of the seed coat, cotyledon and the embryonal axis. The cotyledons of the embryo are simple structure, generally thick and swollen due to the storage of the food reserves. So the cotyledons, based on the cotyledons, so there are two types of seed, monocot and the dicot. If two cotyledons are present, that is dicot, and if only one cotyledon is present, that is monocot. So these cotyledons are thick and swollen, that is because of the food reserve. The seed that may be albuminous seed, non-albuminous seed or the exalbuminous seed. So here non-albuminous seed means which do not contain the endosperm, residual endosperm, that is completely consumed by the seed, that is called as non-albuminous or ex-albuminous seed. And one more type is there, so that is the albuminous seed. So the albuminous seed which have the residual endosperm, they retain a part of the endosperm. So here non-albuminous seeds have no residual endosperm as it is completely consumed during the embryo development. So there is no endosperm is present, for example pea and groundnut. Albuminous seeds, they retain the part of the endosperm. The endosperm is present in the seed, that is not completely used up during the embryo development. For example, wheat, maize and barley, castor are the example for the albuminous seeds. Okay. So based on the presence and the absence of the endosperm, there are two types of the seeds, non-albuminous seeds and the albuminous seeds. So examples are very important. Some seeds such as black pepper and beet, the remnants of the nucellus are also persistent. So this residual persistent nucellus is called as perisperm. What is perisperm? So after the fertilization, the part of the nucellus also present in the seed, that is persistent in the seed, that is called as perisperm. The residual persistent nucellus is called as perisperm. So here you can see the two cotyledons. So this is the dicot seed, and so this is the monocot seed structure. This is the monocot seed, and here in this seed you can see the persistent endosperm. Endosperm that is present, that is the albuminous seeds, and the seed coat. Here can see the cotyledon, root tip and the shoot apical meristem that you can see in the structure of the seed. And here you can see the endosperm. So here also endosperm is present and the cotyledons, and here the different parts of the seeds you can see here. You know that in the seed coat, cotyledons and the endosperm that is present in some seeds, right? So here the integuments of the ovules harden as tough protective seed coats. How these seed coats are formed? The seed coats are formed by the integuments that develop into seed coats. The micropyle remains as a small pore in the seed coat. So if you see the seed properly, that you can see the, so just imagine this is the seed. You can see the small opening, that is the micropyle, that is remaining same as a small pore. So what is the use of this micropyle? So that remain in the seed as a small pore, so that facilitates the entry of the oxygen and the water into the seed during the germination. During the germination, the seed which require the water and the oxygen, so that is uh, the water and oxygen that enter through this small pore. As the seed mature, its water content is reduced and seeds become relatively dry. So when the seeds become mature, the seeds become dry, and which remove its the moisture and the water contents. So then what is the viability of the seed? So generally, when the seed which remove the water content, the general metabolic activity of the embryo slows down. Then the embryo may enter the state of inactivity called dormancy, or if favorable conditions are available, then only it germinate. When the seed become mature, the metabolic activity of the embryo that slows down, means the activity inside the seeds that slows down, and the seed that enter the period of dormancy. Okay, so metabolically inactive state. When the suitable condition, favorable conditions like oxygen, temperature, water, like this, moisture, so that are available, then favorable conditions are there, then the seed that germinate. Okay, as ovules mature into seeds, the ovary develops into fruit. So the transformation of ovules into seeds and the ovary into fruits proceeds simultaneously. The wall of the ovary develops into the wall of the fruit, that is called as pericarp. You know that fruits that develop from the ovary, the wall of the ovary that develop into wall of the fruit, that is called as pericarp. So there are different types of the fruits, fleshy fruits, dry fruits. There are different types, right? So here fleshy fruits like guava and orange, mango. So these are the fleshy fruits which have the fleshy uh, they have the some juicy structure inside the fruit, and sometimes the fruits may be dry. So here for example groundnut and the mustard, they are example for the uh, dry fruits. Okay, then fruits.